So I have nothing to disclose. I just start my talk. So the major goal in epidemiology is to identify risk factors that cause humans to develop disease. But my focus is in the molecular epidemiology. I try to address the question that if under same exposure to why some people develop disease, some people do not. For example, most lung cancer caused by smoke, but only a small portion of smokers will eventually develop lung cancer. So this group of genetic biomarkers that can predict individuals' predisposition to human disease, this is my research focus. So one of the risk factors recently identified, not very recent, but identified is the circadian disruption. Circadian disruption that might increase cancer risk. So this just very briefly highlight our understanding of circadian disruption. That's an internal control. In the morning we wake up, we do not need a cup of coffee, but at night we feel sleepy. That is all controlled by our internal machine that regulate our circadian rhythm, the 24-hour circadian rhythm. That's adaptation of the Earth's rotation, the 24-hour rotation to the sun. The circadian rhythm might regulate hundreds of biological and physiological functions in our human body. And the disruptions of this rhythm have been linked to many human diseases, including cancer. So the majority evidence comes from a shift worker study, but these slides summarize 13 case control studies on shift workers. That's the odds ratio detected. If over 1, which means that's the high risk, less than 1 low risk is the odds ratio. But overall, its combined odds ratio is 1.47, which suggests that 48% increased breast cancer risk among shift workers. Actually, this is the background. This is why I got involved in the circadian gene business, because it's strong evidence support that disrupted circadian rhythm might play a role in tumor genesis. But in 2007, ARC and the WHO, they formed an expert panel to review so far all the evidence so far and conclude that the shift work that involves circadian disruption probably carcinogenic to humans. That's rated 2A. Means the one that's the carcinogenic to human, like tobacco smoke, carcinogen. But this the 2A is very high, but based on sufficient evidence in animals and the limited evidence in humans. So our goal is to provide further molecular epi-evidence using human population to test the circadian gene hypothesis.
So it's okay to drink jet fuel?
Yeah. Oh, yeah. So I don't want, okay. So something happening here. Yeah. So this little off in the top. So speaking of circadian genes, there's a very brief history about circadian genes. The first one identified in the period called period genes, identified not a long time ago, probably 2 decades ago. Then total 9 causes we could call circadian genes. We know many other genes expression regulated by circadian rhythm, but these 9 we call circadian genes actually form the feedback loop control our circadian rhythm. But sequence of all these circadian genes are very conserved. That indicates that their importance in evolutionary history. If we compare the clock gene between mice and the flies and human, probably over 90% of them are very similar. But later on, that's the animal study showing their role in tumor genesis. So this is the simple model I took from the paper. It's the PER2 might interact with P53 and CMYC, as proposed that PER2 might be a potential tumor suppressors. But in addition to this period 2, other circadian genes may also regulate many other cancer-related genes that might play a role in cancer. So this comes our hypothesis to test. So we want to know whether genetic mutations in the circadian genes are linked or associated with cancer risk, or epigenetic changes, for example, promoter methylation, associated with cancer risk, or loss of function, for example, chromosome deletion, chromosome translocation, at the low side with circadian gene might be also linked with cancer risk. So in order to test this hypothesis, we use 3 different approaches. We have a genetic association approach, an epigenetic approach, but all these approaches, we use human population. In the case control study, basically we use blood samples drawn from the patient. We do case control. If, for example, to test the genetic hypothesis, if one mutation is more prevalent in cancer patients, but less frequent in regular normal people, which might suggest that the mutation might be associated with cancer risk. This is how we test this hypothesis using a molecular epi design. We also further identified circadian gene. Since very few studies to examine their functionality, we also try to do some functional analysis, very basic functional analysis to study their role in tumor genesis. So this quickly show the feedback, not positive and negative feedback of all circadian genes, how they manage our circadian rhythm. Total are 9 of them, some of them on the positive arm, some on the negative arm. For example, clock is can promote gene expression with on the positive arm, increase COR2 expression, but increased COR2 expression might also interact with clock protein, reduce clock expression, then reduce COR2 expression. They're very complicated feedback loops, how they regulate our circadian rhythm. So we're going to talk about examples from 2 of these genes, the clock and the MPAS2. So this is one of the key transcriptional regulator, but also the circadian gene, and enhance gene expression. So there's 3 different approaches. As we mentioned in previous slides, we want to know whether mutation in these circadian genes associated with risk, but also hypomethylation in the promoter region associated with risk. So these are all the 9 SNPs we choose in this clock gene. 6 of them in the gene region and 3 of them in the 3' UTR. This the result is odds ratio we find is over 1 with an odds ratio increased risk, odds ratio less than 1, which means that reduced risk is a highlight 1, 2, 3, 4. 4 of them are significantly associated with breast cancer risk. So next slide gives you a little detail. This overall breast cancer risk, I think the total sample size we have is about 450 cases, 470 controls. This is genotype mutation using DNA sample drawn from study subjects. Very interesting. This odds ratio or this risk factor is ERPR dependent. So all of these significant odds ratio detected in ERPR negative tumors. So far, we don't know what happened. Might be that hormone receptor might also be the target of this transcriptional regulator, of this clock gene. Because the multiple comparison issue, we did adjustment. But even after adjust for multiple comparison issue, we still reach significance. So further work is needed to find out what's the ERPR-dependent breast cancer risk. So we also detect methylation level in this clock gene. This is normal distribution in the control, but we can skew to the low methylation level of clock gene. When the low methylation might suggest the increased expression of clock gene. But this odds ratio detected. So again, because the high level of methylation detected in the control sample, this gave the odds ratio less than 1, which suggests that high methylation associated with reduced risk. Low methylation, the increased clock expression associates high breast cancer risk. So whether the clock gene expression altered in breast tumor tissue, so we were able to find a database, that's an array database, public accessible. They have genome-wide expression data, breast tissue from breast tissue, adjacent normal tissue, and healthy control tissue. So as expected from our previous slides, low methylation in cases which might increase clock gene expression in tumor tissue. This is what exactly this data show us. This is the normal from a healthy control, the lowest clock expression, then the adjacent normal tissue, and it's the tumor tissue. It's the highest expression, all significant. So this agree with our previous methylation data. So then we did a little more functional analysis. Since we want, as a transcription regulator, we want to know what are other genes controlled by clock. Then we did the whole genome expression array. Then use the cell line, MSF7 cell line, knock down clock. We find 154 genes with significant expression changes after clock knockdown. And then we did a pathway-based analysis with the network. This is the highest confidence network identified as cellular growth and proliferation, cell signaling, and interaction. So that is the very relevant to human cancer development. So we don't have time to go to detail of the network, but again, this further evidence suggests possible role of clock in cancer development. So the second gene I'm going to give example is MPAS2. This is another circadian gene, also is the biggest circadian gene. There's only one mutation, very conserved. The whole sequence is very conserved. Only 174K. Only one mutation that causes amino acid change. So we only genotype this one because the rest of the region is very conserved. We genotype it in several different cancer types. Also perform functional analysis. I'm going to show you brief data what we got. So this mutation, we call the missense mutation, the amino acid change. We get odds ratio, significant odds ratio 0.41. We suggest that people carrying this mutation have a 39% reduced breast cancer risk. So we find in both postmenopausal women and premenopausal women. So overall, very significant is breast cancer, but the same mutation also show association with reduced risk of non-Hodgkin lymphoma. So again, odds ratio 54, 0.55, which means that 45% reduced risk. So it's consistent with the finding in breast cancer study. Then we were able to test whether this mutation and expression of the MPAS2 could be a biomarker for breast cancer survival and recurrence. So that's the genotype. We find the in overall disease-free survival, there's significant correlation, but also expression, high expression of this MPAS2 gene associated with overall survival and disease-free survival. Because high expression, we suggest that this gene, in addition, in contrast to clock, might be a tumor suppressor because high expression give us benefit of survival. As a transcription regulator, we want to know whether some cancer-related pathway genes might be affected by MPAS2, by this circadian gene. Then we first we try this DNA damage pathway gene, total 84 on the plate. Majority of them, after knockdown, majority of them decrease, this is downregulation. This is consistent with the enhancer role of the circadian gene. But further, we want to know what are exactly target of this transcriptional regulator with the whole genome chip-on-chip analysis, identified 104 direct target, but of those in 10 are also transcriptional regulator, and 9 of them are genes with known role in tumor genesis, including P16 intacting protein. Let's show the brief result. This the P16 intacting protein suggests that this MPAS2 might regulate other cancer-related genes and play a role in human cancer. Then we further do some phenotypic analysis. So that's DNA repair, cell cycle, apoptosis, the 3 major cancer-related biological pathway. So if we knock down this MPAS2 in cultured cells, then we use mutagen to challenge the cells. What happened? So this is what happened is the common assay we perform is a longer tail. This tail is DNA break. The longer the tail, which means that the high level of DNA damage. So this is the MPAS2 knockdown cell line with mutagen treatment give us longest tail moment. The tail moment which means the highest level of DNA damage, which means that suggests that MPAS2 might help or might be involved in DNA repair pathway. So this give an overall results we found so far. The MPS2 clock, there are 2 examples we just mentioned. But total, the 3 different cancer types we used. The breast cancer, this is the case number, control number. 6 of circadian genes have been found to be associated with breast cancer risk. But very interestingly, all of the circadian genes have been identified to be associated with prostate cancer risk. This is also very large sample size, over a total of 2,500 case controls. But also non-Hodgkin lymphoma, 3 of them of circadian gene have been identified significant associated with non-Hodgkin lymphoma. So again, we just start doing functional analysis, try to figure out detailed role, probably different pathways might be affected. But if we go back to the very original slides, shift work increase cancer risk. And the circadian gene, to summarize what we found from our epigenetic association analysis, circadian gene are associated with cancer risk. But still there's the gap between shift work and the molecular changes. Now we just began to find out whether any genetic and epigenetic impact caused by shift work. So this is to summarize the methylation results from case control study. So clock gene, for example, we use the clock gene. Clock gene is lower methylation, which means the clock will be highly expressed in breast tissue. But whether low methylation could be caused by shift workers, long-term shift workers. So by collaborating with our colleagues in Denmark, there's the Danish cohort, we were able to get this hypothesis tested. So the long-term shift worker, this is the day worker. In long-term shift work, we can see that this is a low expression of clock. About 58% of long-term shift workers have low methylation clock promoter compared to day workers. But if you can remember, the low methylation of clock is seen in breast cancer cases from our case control study. These two are great each other. This might indicate one of the first evidence linking possible mechanism that might cause cancer by long-term shift workers. So now we try to do the rest of circadian genes, but also do the global methylation pattern changes to see whether any other loci affected by long-term shift worker. To conclude my talk, I would thank my lab group. Aaron Hoffman did most of this analysis, and Dr. Townsend provided a breast cancer, non-Hodgkin lymphoma, case control study samples for testing a hypothesis, and Herbert, and also collaborators in Denmark provide samples for testing long-term shift worker hypothesis. Okay, thank you.